Hey guys, Contino Academy here with another biology video, and today we'll be talking about this thing called mitosis. So, what is mitosis? Well, basically, as we've talked about before, cells make up all living things, and they die, so we have to have a way to get new cells, or else we would die with ourselves, which eventually we do, but not immediately. So mitosis is how we create new cells, and it's a process. It's split up into different stages, which we'll talk about more in a second, but I'll just, so you know these phases, just type them out for you. Here we have interphase, prophase, metaphase, anaphase, and telophase. So there are these 5 phases that make up mitosis, and they occur in this order. The order is important, and the way I remember the order is IP on mat T. It's disgusting, I know, but that's how I remember it. IP on mat T. Interphase, prophase, on, metaphase, telophase. IP on mat T. So now let's talk about each one individually. But first, as I've said, it happens in cells, so we need a cell to look at here. So there's a cell, and more specifically, we need to look at the nucleus of a cell, because it all happens in the chromosomes. That's pretty much what mitosis is focused on, chromosomes. So let me just draw this out here. So anyway, in a typical diploid cell, we have 23 chromosomes. So I'm only going to draw 2 because 23 is a lot of little lines to draw. So there's really 23 of them, but I'm only going to draw 2. So this right here is what a cell looks like before it starts mitosis. And then it begins mitosis with interphase, which is here. We'll make this cell be interphase. And what happens is in interphase is it starts prepping. It starts getting ready to divide. And the main thing that occurs here is it duplicates its number of chromosomes. Because right now there's 23 chromosomes, which are in the form of chromatids, technically not, but we're going to call them chromatids. And then in interphase, it duplicates. So now there's 46 chromatids, but only 23 chromosomes. Because as we've talked about, one chromosome can be made of 2 chromatids. So here's the before. Before interphase and after interphase. So here we have 23 chromosomes, just single chromosomes, and here we also have 23 chromosomes, but they are composed of 2 chromatids, identical chromatids. It took this and it directly copied it right here. It took this and directly copied it right here, 23 times over. So that's interphase. Now let's go to prophase. I'm just going to copy this whole thing down so I don't have to redo it all. That arrow shouldn't be there. So what happens in prophase? Well, just draw that arrow down here. That's a really bad looking arrow. Anyway, so before prophase, it looks like, well, the after phase of interphase, because after interphase comes prophase. So this is what we have. We have one cell with 23 chromosomes made up of 2 chromatids each. And what happens during interphase is the nucleus of the cell actually starts disappearing. The nuclear membrane starts breaking up. The nucleus is essentially, just like I said, disappearing. You're losing your nucleus. And at the same time, you're getting these things over here called centrioles and spindle fibers, which I'll talk about in a second. So what are centrioles? Well, these things right here, these are the centrioles, the base. They're just bases, 2 on each side. Well, 1 on each side, 2 total. Bases where spindle fibers come out of. So this right here, I'll make it a different color. This is a centriole. This is another centriole. And then these red things come out of them, and they're called spindle fibers. And what they're eventually going to do is they're actually going to attach to our chromosomes. They're going to latch onto them. But that happens later, so we'll talk about them more later. Centrioles are the base that spindle fibers come out of. So, to review so far, Interphase, before 23 chromosomes, after 23 chromosomes, each made of 2 chromatids, so it made identical copies of its chromosomes. Prophase, before, like we just said, 23 chromosomes, each made of 2 chromatids, and after your nucleus is starting to dissolve, you're getting centrioles on each side of the cell and spindle fibers. Metaphase. So I have to scroll down here to get some more space, but metaphase. Here we go. So during metaphase, the chromosomes, the nucleus is completely gone. There is no more nucleus. Consider it gone. Poof, gone. And the chromosomes, that 23 chromosomes, I only drew 2, but there's 23, they actually line up in the middle of the cell. That's what they do in metaphase. They just move on down to the middle of the cell, all 23 of them. And nucleus is completely gone, like we said. The spindle fibers and centrioles are continuing to grow. Let me find the color. I'll make centrioles this color because I don't want it to be the same color as that chromosome. So centrioles right here, and spindle fibers are actually starting to attach to the chromosomes. They're reaching out and attaching to the chromosomes, all 23 that are lined up in the center. This is happening during metaphase. I'm going to get rid of this before and after because it's redundant. Because the before of one is the after of another. So this is metaphase right here. And since the text is moved out of the screen, I'll just label it metaphase. And let's do a quick review. So interphase. Before interphase happens, we have a cell with 23 chromosomes after it copied each of its chromosomes identically into 23 chromosomes made of 2 chromatids. Prophase, the 23 chromatids, the nucleus is starting to dissolve, the centrioles and the spindle fibers are beginning to form. Metaphase, the nucleus is completely gone, the chromosomes, all 23 of them, line up at the center of the cell. The centrioles and spindle fibers are beginning to attach to the 23 chromosomes. Let's move on to anaphase. So what happens in anaphase? Well, in metaphase, as we said, the spindle fibers are attaching to the chromosomes. In anaphase, they actually rip the chromosomes apart into their chromatids. They take the chromosomes, the spindle fibers do, I'm talking about right now. When I say they, I mean the spindle fibers. So let me draw this out. In fact, let me zoom in. Not that far. So in anaphase, the chromosomes have actually been ripped up into their chromatids. So we have one over here, and one over here, and one over here, and one over here, and the cell itself is actually starting to spread apart. It's starting to divide. It's spreading away from itself. Like this. Excuse my artistic terribleness. It is the best I can do. Fill it in. And that is anaphase. So in anaphase, the chromosomes have actually been ripped apart into their sister chromatids, which are now called chromosomes when they're by themselves. Because, as we said, you know what, I feel like I should review this because it is confusing. So, here is a chromosome. Well, that's what a typical chromosome, right here. And it is made of 2 chromatids. This right here, that's a chromatid, and this right here is also a chromatid. So chromatid and the whole thing is called a chromosome. But because biologists want to be confusing, this is a chromatid. Like this, I just drew it down here. When it's by itself, it's called a chromosome. When a chromatid is by itself, it's called a chromosome. And 2 chromatids together, if you do that, they're still called a chromosome. So this is a chromosome, and this is also a chromosome. But this is a chromatid. So one more time. Right now, this is called a chromosome. And now, this, my bad there, this is called a chromatid, while this entire thing is now called a chromosome. So quick review, this right here, chromatid, this, chromosome, this also chromosome, which is a chromatid, but chromatids by themselves are called chromosomes. Alright, so that's anaphase, and that's a little review there. Now let's move on to telophase. I'm going to copy this over. I have to go find it now. And there it is. Alright, telophase. Telophase. Alright, so what happens in telophase? Well, the cell keeps pulling apart. It's getting further away. It's dividing even more. That's probably enough. And then I gotta draw this in again. I should be an artist. I'm like the next Picasso, aren't I? I think so. Fill it in with black. And whilst this is happening, there's a distinct line of like division. They are clearly dividing, and a new nucleus actually begins to form on each side of said line. So over here, where the centrioles and the spindle fibers were, a nucleus is beginning to form. It's not completely formed, it's just beginning to form. So it's forming, it's kind of getting there, so I'm not going to fill it in all the way because it's not completely formed, but it is forming, and the centrioles and spindle fibers are disappearing. So nucleus is forming, centrioles and spindle fibers are going bye-bye. Alright, so those are the phases, and the final product, what it looks like at the very end, is, this is actually going to be a much easier way to do it, is right here. And this is sometimes called cytokinesis, sometimes it's just called the end of mitosis. So they are the same thing, cytokinesis and the end of mitosis are the same. And this is what it looks like. You have two complete new cells with the exact same, what's the word I'm looking for? Chromosomes. Wow, that was terrible. The exact same chromosomes. And remember, they have 23 chromosomes, which are not paired at all. They're just by themselves. Like here, they're just like this first cell. This is what they are at the end. You have two of these, just single chromosomes. Right there, boom, which is sometimes called cytokinesis, like I said, or the end of mitosis. Cytokinesis. And that is mitosis. And then this cell right here is going to do the exact same thing that happened to this cell. It's going to go through the exact same process. And this cell right here is going to do the exact same thing that this cell right there did. Over and over and over again. So, quick review. Mitosis is made of interphase, prophase, metaphase, anaphase, telophase, and cytokinesis, sometimes called that, sometimes just called the end of mitosis. So IP on mat C. That A I really should have made capital to emphasize the anaphase. So IP on mat, parentheses C. It should be a rap song. IP on mat, parentheses C. Anyway, interphase. The cell has 23 chromosomes, and it copies them into 23 chromosomes, but 46 chromatids, as we talked about here, the whole naming issue. In prophase, the nucleus starts to disappear, the centrioles and spindle fibers begin to form. In metaphase, the chromosomes, 23 of them, line up at the center, and the spindle fibers actually attach to the chromosomes. In anaphase, the spindle fibers pull apart the chromosomes into their chromatids, which when chromatids are by themselves are then called chromosomes. So we have 23 chromosomes here in metaphase, pulled into 23 chromosomes over here, and 23 chromosomes over here. In telophase, the spindle fibers and centrioles begin to disappear, a new nucleus begins to form, the cell gets further and further apart, and in cytokinesis, or the end of mitosis, you have two cells that look exactly like the beginning cell. And that is mitosis. If you have any questions, leave me a comment or check out biozol.com.